to thank everyone for joining today's session. And we will be presenting the consumer presentation today with the vaccines and with Professor Alex Sikorski and Dr. Michael Babbage. The agenda is we'll start with at 12 o'clock sharp with the presentation and overview of the consumer IEEE Consumer Technology Society introduction. And then we will run through the presentation, and at the end of the presentation, since it's a webinar format, you can ask your questions. Everyone is unmuted, meaning you can be unmute yourself and talk and ask questions at the end to make the presenters more comfortable. On that note, allow me to start. So please post pictures, tweet, post concerns, and etc. The officers for 2021 in the IEEE is yours truly, and Dev Bacharaya, who is the co-chair, Avik, vice chair, Pat Kasank is our secretary and treasurer, Michael Wang is the marketing chair, Joseph Wei is chair of the consumer electronics and also part-time webmaster, and Palak Talwar is marketing co-chair. We are always looking for additional volunteers, and 2022 will be the same. If you have ideas for presentations and workshops, please send it over to me and we'll work accordingly. In December, we'll have another speaker. We have right now several of them line up, so we'll see who's presenting in January, who's presenting in December. You, as always, please become a Consumer Electronics CE member or IEEE member. It's a great organization with a long history and high quality content. You all know the presentations and standards are developed by the IEEE engineers. We have a magazines like Consumer Electronics and IEEE magazines, so you get a free subscription. If you have topic suggestions for 2022, please send it over. And if you have any job opportunities you would like to share, please share it with us so we can distribute it to our list. On that note, allow me to start with the speaker introduction. The speakers are Alex Sikorski and Michael Babbage. Alex is the founder and president of BioVirus Research and bioinformation mathematician, geophysicist, software engineer, and military contractor. Actually, Alex has been in Afghanistan and a lot of missions. He has several US patents in the fields of virology, immunology, and genetics. Alex has over 40 years experience in gold and oil exploration, biotech, and tech industries, ranging from full cycle development and patent and synthetic universal peptide vaccine candidates to instant thermoplastic holographic imaging cameras. He works with the Department of Defense, West Point Military Academy, CIA, NSA, you name it, he probably worked with them. Pentagon, quite interesting. And he worked for 2.5 years with the Deputy Undersecretary of Defense, Paul Brinkley, in Afghanistan. Our second speaker is Michael Babbage, and Dr. Babbage is co-founder and CEO of LabRx Immunological Therapeutics, where he designed and led a cancer vaccine for FDA approval for clinical trials at University of California, Davis. He has over 35 years experience to include faculty positions at the University of Illinois, UC Davis, and co-founder of multiple startup biotech companies. Prior to biotechnology, he served in the U.S. Army Medical Service Corps, directed hospital with international operations. Dr. Babbage has over 100 years combined papers and research and grants. He completed his postdoctoral training in endocrine unit at University of California, San Francisco, and learned his PhD in pharmacology from University of Kentucky. And the topic, which is near and dear to my heart, and I hope you all will concur to that. It's a vaccine technology. The team worked on it quite a bit, so we're bringing it today. And they also have an IoT angle and consumer applications as well. So that's why we're presenting today. So vaccine technology has made a lot of progress in 225 years since Edward Jenner and Catherine II took the innocular practice of Turkish military doctors to public use. Lots of innovation were adopted after the time. And the core algorithm still remains the same, as you can see with our booster vaccines. The established vaccine technology just mimics the natural process. Number one, its adaptive human immune system is presented with and sees the most prominent external part of invading virus, so-called epitopes. These are the viral surface proteins like corona S spikes on the surface of coronavirus. We all know what that means. That prompts the uniquely specified immune response antibodies. The protein against which the immune system issues its response is called antigen. Today, practically all antigens are viral epitopes, and there lies the problem itself. The process is very specific to the viral epitope, and thus the resultant immune response is uniquely specific to the given variant of the virus. This nature mimicking approach worked well for a long time for genetically stable DNA viruses, but has some serious problem with fast mutating RNA viruses, whose epitopes quickly mutate and render the immune response less and less effective, as proven by current pandemic. Since we cannot do anything about making RNA viruses less mutative, we are mutating nonstop. We need to abandon our nature mimicking epitope-centered vaccine paradigm and enter the new approach in vaccine design, in antigen engineering for higher vaccine universality. The team will present today two, but both highly universal vaccines, both proved in vivo and protection trials, one already in FDA trials and against many adenocarcinomas, and the second potential opens a whole new class of viral family-specific vaccines. The last one addresses the very resistant viral family, herpes viridae, that include many herpes viruses, from genital virus to Epstein-Barr, cytomegalovirus, and Kaposi sarcoma, and more. On that note, I would like to exit my presentation, stop sharing, and introduce one more time Alexey Horsky and Michael Babbage. And you are all welcome to ask questions through the Q&A, and at the end of the session, we'll address them. I will promote everyone so you can speak once you unmute the phone. Please, Alex and Michael, thank you. And Alex, I would like to start with you and with your presentation. And I want to thank you for presenting today.
Thank you so much. I will, yes. This is what we will try to do. Give me a second. I'll see it so I see myself too. Okay. My name is Alex Chehorsky, and thank you so much for everybody to take part in this. This is my colleague, good friend, and a competitor in a way, Dr. Michael Babbage. We've known each other for a long time because both of our companies were basically working for a quarter of a century each on a pure enthusiasm and for very few people paying attention at what we do. But now that we have a pandemic upon us, suddenly what we were doing for all these years is starting to make sense. But for it really, and of course, everybody hears today everything about virology on TV and on radio, but I still would like to spend a little bit of time coming back to terminology and make sure that we're talking about the same things. It is very easy for us to get off the straight line of a straight logic and start getting into the depth of virology, which is completely endless. So I will try to stay on that ridge and not get into something that may really be exciting for the specialist, but is not that interested for general scientific and engineering public. And we'll get back to the line of my story that will allow you to get more excited about what we do and understand the core of what we do, rather than the details, which you can ask questions later or read about later. So let's get back to the basics. Everybody saw the pictures of the virus everywhere. We will be using kind of like a model that I use for the coronavirus, which is a round shape with these spikes on the end. And what the virus uses these spikes for is that when it contacts with a cell, they play a role of keys. They open up, so to speak, the cell, the membranes fuse, and the virus drops its contents into the cell. And since that moment, cell stops doing what it was normally doing and start doing what a virus program makes it do. That spike on the surface, I made a little model of it with the Lego. It doesn't really look how it looks in the nature, but it's good enough to discuss the properties. The name for this is antigen, and you don't hear it's a superficial protein on the surface of the virus, and it plays, as I said, as a key for the virus to open the cell. But on the other hand, this is what immune system sees, and this is how immune system determines that they have an invader. So basically antigen, and it's not a good word, it has nothing to do with genes. So antigen is not a very explanatory term on its own, but we're all used to that, and everybody else will have to get used to that. So this antigen is how the virus, the immune system sees the virus. So if you think about your immune system as a computer that has to do something about the invader, that is how it sees the virus. And basically the immune response, so what your computer spits out to fight, so to speak, that virus, your immune system consists, is a reaction to that antigen. So when we do the vaccination, the vaccine is actually antigen plus that's the method of its delivery to your system. And of course, the most important is the message itself, the antigen. The delivery takes the second place. You can deliver your message to your computer through floppy or through the internet. It doesn't matter. The same thing with the immune system. Somebody can sneeze on you and deliver the virus that way, or you can get an injection as a vaccine. The immune system does not know any difference. So the immune response, the output of your immune system, is almost completely depends on the information it reads from the antigen. And the consequences of that is that somehow the immune system, it's a very complex process, how it figures out how to fight with that, what it issues, what is the immune response is. We will discuss today only one side of it, which is the antibody that everybody heard about, is an antibody. An antibody, and I will have another model for that, is a Y-shaped, small, tiny, I would say, protein. And what it does is that, since you remember, we have that virus and an antigen on it, right? So what the immune system does is that it clings to the surface electrostatically, electromagnetically, whatever you want to describe it, the protein to protein chemical and energetical connection, it clings to these things by the special proteins that is always different from a different from virus to virus, because that is how, by recognizing the antigen, it issues these specifically to cling to them. And that way the virus cannot touch the cell anymore. It's physically between the virus and the cell. So this is how, in 3 minutes, if you want to describe virology and virusology in 3 minutes, that's what it was. So the importance of antigen is basically the core of the whole immune system work. And as of today, interestingly enough, all the innovation, or most of innovation, let me put it this way, were put everywhere in the vaccine, in the delivery system, in the special additions to the vaccine that makes them more active, but not in the antigen, not in the most important, in our opinion, part. What happens is that we're still mimicking the nature. And by mimicking the nature, what I mean is that we're delivering to the immune system either the virus with that antigen, or that antigen separate from the virus, delivered some other way, like the mRNA vaccine. mRNA is a delivery system. It delivers the same antigen that is being delivered in your system by somebody sneezing on you. It's more effective. It's much cheaper to make the vaccine that way. It is much more stable. There's much less problem with the mutations during the manufacturing of the and everything. But still, it is the same antigen. And there, the big problem happens. Because we were using the vaccines for more than 200 years. And all that time, the core of the vaccines remained the natural antigens. And we were mimicking again, the nature that way, but it all was good while majority of the virus that we were addressing were DNA viruses, because DNA viruses are not that ready to, I mean, they mutate, but not as easily as RNA viruses. So when you made a vaccine against a DNA virus, it remains stable for the very, very long time. With the RNA viruses and the flu you can use as an example, it's completely different. It mutates so fast that these small things on the surface of the virus are changing fast. And the vaccine that you made a year or 2 years or 5 years ago may or may not work with the new mutated virus. And what we see today on the coronaviruses is that even a faster process. When we have new viruses, new variants of the viruses are coming every half a year or a year. And of course, when this is all started in 2020, majority of the virologists knew that this is what's going to happen. So a little bit they were thinking that, okay, we're going to make boosters, or we're going to put more of this of the new antigens in a vaccine. But again, it's getting faster and faster. And in our opinion, for over 20 years, we were thinking that the science of vaccinology should be aiming to creating more universal vaccines. What I mean by that is vaccines that cover more strains, more variants, and preferably other viruses in the same viral family. And in that case, you will be protected not only from new variants, but also from non-yet existing viruses that were not yet discovered, as long as they belong to the same viral group. And it looks like a dream, but actually it is not a dream, because we're here to present you 2 vaccines that we developed during the years. Mike's company developed the vaccine against all known adenocarcinomas. And adenocarcinoma is a cancer that is about 45% of all cancers, but that 45% are responsible for about 75% of all cancer deaths. So basically their vaccine, if it does what we hope it do, and it looks like it does, it's now in the first stage trials of FDA. So FDA allowed it to be inside the process, which is, for those of you who are not familiar with the biotech and all that, it's a huge, huge, huge achievement. And the vaccine that I was developing with my guys for the also past 25 years is a vaccine against at least many, we cannot say all, because it was impossible for us to test with all, but with many different herpes viruses, both human and animal, which is important because a lot of agricultural products are like chicken are inoculated against herpes virus. Otherwise, you would not even touch them. And the human herpeses, which include genital herpes and other herpeses, Epstein-Barr, I will be talking after Mike will introduce their vaccine. I'll be talking a little bit more about our vaccine as a product. So instead of giving you all that scientific background and then about the results, I'd rather start with the results, with the two vaccines that exist today. One of them, again, is in trial. And if it will be fulfilling its promise, it will be both preventive and therapeutic, meaning that it will prevent you from getting cancer. But if you got cancer, it will also be treating that cancer. And we're talking about 75% of the killers in a cancer field. So so how we do that? What is the main, where we are common? We're common in the ways of, we completely reworked the concept of how to make the vaccine because we're not using the whole antigens. We came from that, we came from the mimicking nature to the engineering, meaning that instead of just doing what the nature does, we're trying to find out how small things are happening and try other things. So then we're building not a bridge like a tree across a river that a Neanderthal man saw and used as a bridge, but a real bridge that we want to engineer according to every time we deal with another type of the virus and a vaccine, we engineer it that way. So the hope is that we will be able, using our technology, and it's different technologies, by the way, because Mike and his guys, they're using biological technology, so to speak. So they try to find the areas in the virus that are common, that are more conservative, meaning they mutate less, and create a vaccine that would cover them all. What I am doing, and what my company was doing, is that we use pure math. We go into the very, very interesting algorithms, and we are trying to find algorithmically the structure, a small protein structure that will be either common or by some other way that will deliver the antigens, the antibodies to that formula, will deliver the protection against all viruses in that group. And believe me, the herpes virus group is one of the most difficult groups because there is still no vaccine today against, for instance, genital herpes or Epstein-Barr herpes or cytomegalovirus. And it causes a lot of damage because, for instance, cytomegalovirus leads to the deformities in the newborn. And there's about 100 million active genital cases in the US alone. And in the world, there's hundreds of millions of people who suffer from both more or less a common herpes, which when you have the sores on your mouth, and the genital herpes, which could be very debilitating. So what is it we do? Instead of using the whole big proteins, the spikes, the antigens, we make antigens out of tiny proteins, which again, we calculate, and these guys find it biologically. It was, for all these years, it was always mostly rejected by the big science that such small proteins, we're talking about 20, 30 amino acids, and actually they can be smaller. Small proteins can deliver that kind of a message that would cover these many strains and many viruses and many cancers. But actually we proved that it happens. It did not help us much with the recognition in the general science of virology that we do, because for different reasons, but mostly probably because it's so far away, as a beginning at least, right? Yeah. Was from the thinking in the science to a
dogma.
Yes, on a dogmatic side, but that can be debated. But one way or another, what we do now, the Mike will tell you about their vaccine, the cancer vaccine, a little bit more. Basically, you got the most information already, more or less, but he will tell you more about their vaccine. And after that, I will tell you a little bit more about the herpes vaccine, and we will conclude with the telling you how this can be used in a COVID on our RNA viruses situation, so that we will be not only protected, but what we have now, but we will be protected from other variants in the future, already protected, because the universal vaccine, again, covers not only the existing viruses, but only known viruses, but also the viruses that was not discovered. And I'll give you one example of this. So I hope it was not, my delivery was not too fast. And if you have questions, we can address them later. But for now, Dr. Babic, this is your.
Good timing.
Yeah.
Thank you, Alex. What I'll be describing is our novel cancer vaccine, as Alex alluded to, and our engineering approach to both prevent and to treat existing cancers.
Oh, wait a second. I need to share. Right?
What I'm going to do is share the PowerPoint for you, and Alex is pulling that up.
Is it shared?
That's good. That's good. And then I'll just click that one right there.
Which one? This one?
Mm-hmm. Mm-hmm.
Okay.
Although, is it showing up on your screen?
Yeah, it says stop sharing, so that means that it's shared.
I just want somebody to confirm that they're seeing the full screen, because we're not seeing it. It looks like we've
Yeah, we see the full screen. It's all good.
Okay, thank you very much. Thank you. So what I'd first like to do, moving on with this, continue on with the story with the cancer vaccine as an example, I think it'd be good to give you a little bit of the scientific experiment, little background to give you some context of where this is coming from and how we're a little different from what has been 200 years worth of pretty much common thinking, and where we deviate a little bit differently and innovate slightly differently today between our vaccines in development. Edward Jenner did an experiment and observed that milkmaids that contracted cowpox didn't get smallpox. He concluded that somehow there was some protection being developed in the body against any pox. So what did he do? He scratched some of the pox from the milkmaid onto the boy. I don't think there were too many committees regulating human studies back then. So he did that study. And then once the boy recovered, he scratched into the recovered boy some smallpox onto his skin, and his protection was complete. So what is this? What did he learn? What's the take-home message of the discovery? This experiment by Jenner demonstrated that the body makes some substance that defends you against whatever causes the sickness, a virus in this case. The amount of infectious material to administer, of course, has to be in the right amount. You put too much in and you infect the person and they get sick. You don't want a level that's really high. So you bring it down to just enough for your immune system to detect it, but not too much to be clinical, meaning you're going to get sick. That way it wakes up the immune system, if you will. And what we now know are primary antibodies being produced, as well as other significant pathways we won't go into at this point, but there are several other pathways, but the antibodies are the primary ones you've heard about. Since that time with Jenner, of course, we know about vaccines that are preventative vaccines, but there are no therapeutic or treatment vaccines. That is, if you're already infected, the vaccines today, they're not going to help you. They're going to help prevent you from getting infected, not to act as or serve as a treatment. And furthermore, with respect to cancer, there are no vaccines approved. What's the problem? Well, usually a cancer cells can trigger a response on their own so that the person's immune system destroys the cancer before you can even detect it. In some people, their immune system, for some reason, does not recognize the cancer cell as a foreign object. They think their body thinks it's part of their natural genetic makeup. However, with the discovery of a cancer marker called labyrinthine, we can engineer antigens for not only to be used as preventative vaccine, but also as a therapeutic treatment vaccine. We can do that because labyrinthin's common to many cancers called adenocarcinomas or glandular, and that's where they arise from. And it's conserved, meaning, as Alex alluded to earlier, it doesn't mutate or change. So it would be consistently available as a target for both preventative and therapeutic vaccines. That has led to the recently FDA-approved vaccine we have called LabVax to go into clinical trials at the National Cancer Institute Designated Cancer Center at UC Davis. It contains 4 proteins, 4 small protein chains derived from the labyrinthine structure. And the idea is to raise your immunity against labyrinthine. So that is, these 4 antigens were designed to stimulate a person's immune system against labyrinthine, and thereby, as a consequence, any cancer cell that displays labyrinthine on its surface will be detected and attacked. So why is this approach unique? Well, first of all, as I said, there are no therapeutic vaccines across the board, let alone in cancer. And it's a therapeutic vaccine, but it's also a vaccine not just for one particular type of cancer, but a broad amount, or a pan. You might hear pan adenocarcinoma target is labyrinthine, or a pan vaccine in this case. So it's a broad group it can attach. So adenocarcinomas are glandular. Anything that secretes something, a hormone or some juices or something. So we have breast, stomach, ovarian, prostate, lung, pancreatic, colorectal. Those are the primary ones that make up the 45% of all cancers. There are a few others embedded in there and a smaller percentage. Esophageal cancers, there aren't too many in the first place. And of those, probably about 15–20% of esophageal are adenocarcinoma. For example, when you swallow, you notice it's lubricated to allow you to swallow. So that part of your esophagus is, some of those cells are glandular. So it's included in this group. And if you just read across the board, most of you will recognize that these glands and these sources are really generally deadly. Pancreatic, obviously, being a very key one. Ovarian in late stages, very small percentage of survival rate. So what this says is it also changes our thinking in that this is we can now do multi-site diagnosis and multi-site treatment from those sites you see up above. For example, even every time I say, well, we're developing a cancer vaccine, they say, well, what type? And they're expecting me to say breast, stomach, ovarian, prostate, lung. Well, traditionally is breast or lung or pancreatic. But by having a novel target like labyrinthine that's associated with all of these cancers, and also I'd like to mention it's not been found on any normal cells. So this is very specific. You'll have less side effects, it would be the prediction. It's very specific. And it's also not on other cancers. It's just very unique to the glandular or adenocarcinomas. So basically, we could have a treatment for breast and lung and pancreas, et cetera, et cetera. Now, you may only have a lung, not you, hopefully nobody in the audience. I'm not referring to you, but a person could have a lung cancer or pancreatic cancer, but very often it becomes a metastatic, and so it becomes among these, you have more than one type of cancer. So ours is approaching it as a pan cancer treatment across all if they have one or more of these, that's what the idea is. In short, the idea is that as cancer cells with labyrinthine arises, they're now seen as foreign agents, foreign to the human, to the person or the patient. Labyrinthine it's also unique in that it offers several antigens to engineer as a vaccine. By having many antigens, you increase your probability of getting a family of antibodies to attack the target. The idea is that as the cancer cells with labyrinthine specifically arise, they're now seen as foreign, and very sensitive mouse here, seen as foreign, and they display labyrinthine, and due to the 4 engineered antigens that are contained in the labyrinthine vaccine, the patient is expected to produce a family of antibodies against all 4. So as labyrinthine arises, they're now seen as foreign and destroyed by the immune system. So to look at it another way, let's compare the available COVID vaccines in general, schematic terms, how they work versus what our approach is. So you have the SARS-2 for short, if you will. This is the virus, and Alex showed you the toy model of a virus. And it injects its damaging genetic material into the host or human cells. And the injection part that binds with our cells is called the spike protein. So it's on the surface of these cells is the spike protein. It literally inserts itself into the host cells, and then it injects its demon RNA or DNA into you and does its damage, and you become ill. So vaccines have been developed that expose the body to the spike protein. The developed vaccines contain either the manufactured spike protein itself to be injected, like what Jenner did with the cow and smallpox those years ago, actually reproducing or injecting the primary substance itself. Or the other methods to introduce the spike proteins are very indirect, as Alex mentioned, and you've heard about these mRNA or messenger RNA vaccines that, in short, they just code for you to produce the spike protein. So they inject an RNA that in turn you make the spike protein to, and that is the antigen for you to produce antibodies against that target. By comparison, when a normal glandular cell becomes cancerous, it displays a new marker target. This is the labyrinthine. Labyrinthine exposes the patient to these 4 protein sections to be antigenic, as depicted here by the little red stars. So labyrinthine is the entire length of this, but we just picked out little pieces there that are the antigenic pieces. And even though labyrinthine is common to all adenocarcinomas and conserved, it maintains its structure, for example, there's less chance for it to mutate and lead to resistance to the vaccine treatment. That is, even if an unexpected mutation does occur, and our preliminary data suggests it's very stable, but if it does happen to, there are 3 other antigens available to raise antibodies and immunity against labyrinthine in the cancer cells. So the take-home point to compare these two is that the SARS vaccination is somewhat classical, even though there's some modern approaches with the mRNA vaccines, but it still in the end leads to a classical vaccine, and that they're designed to protect against an invading virus and thereby prevent the COVID-19 illness that results from SARS-2 infection. Whereas LabVax, on the other side, is designed to stimulate a cancer patient's immune system against an already pre-existing target, labyrinthine. And that's your difference. Alex?
Yes, thank you so much. Let me do this. Yes, so we're gonna kill this thing. And so let me talk a little bit about herpes vaccines. I really don't want to make take too much of your time, because everybody knows what herpes is. And our vaccine was, I can show you for the people who are most, oh, sorry, not this thing. DARPA, the one, this is from our presentation to DARPA. Not that it was very successful as a presentation, but I want you to see, and we share that again, right? Here?
Alex, can you zoom in? Yeah.
How about that?
Perfect.
Okay, so this is for people who are a little bit more interested in the process. So how do you know that you have a vaccine? First, you come up with some antigen, whether it's the big antigen like we were talking about, or our tiny antigens. You inject it in, let's say, rabbit, okay? And then you let rabbit develop the antibodies. You take the blood, you separate antibodies, and you test antibodies in vitro, meaning that there are a series of tests that allow you to see whether your antibodies are active against that virus. There are a cheaper one, which is that ELISA titration table, and there is a more expensive one, which is neutralization. But one way or another, if you look at that yellow, ELISA titration table, you will see that each of the antibodies, they're called anti-P1, anti-P2. P1 and P2 is our peptides, is that the tiny antigens. And the antibodies to them are anti-P1, anti-P2. And on the horizontal scale, you see different human and one of them chicken herpes viruses, which is HSV1, HSV2, which is general herpes, cytomegalovirus, Epstein-Barr, varicella zoster, Kaposi sarcoma herpes virus. That virus was discovered relatively recently in the 90s. And what's interesting about that is that we had a vaccine already by that time that was working and that was manufactured by us. And we tested it with a Kaposi sarcoma, and it was active against Kaposi sarcoma, meaning that we had the vaccine against a not yet even known virus at the moment. And the MGV. The higher the number, the more active the vaccine is. This is a relative numbers, but these are the dilutions, how much you can dilute the antibodies so they're still working, so to speak. Then the green table is a table of neutralization, where you're actually neutralizing the virus itself, which is also against several viruses here. And after that, you move into the most important part. And the most important part is in vivo tests. So first, you learn from in vitro that you have chemistry. It does work. But then you need to know if it works at least in animals first, before you move into humans. And we had two, I never do anything in our labs, and we don't maintain one for that reason. We always use the professional people at the universities. So we were testing the same vaccine against human genital herpes in McMaster University. And that same vaccine, together with ours, were used against chicken in a MDV. Why? Because that's a very good target for a quick development of a vaccine. It's not a human vaccine, so you don't have to go through FDA. You only go through USDA. Billions of chickens are inoculated every day, so to speak, with that vaccine. The traditional vaccine is very unstable. It's very difficult to use. Our vaccine was very successful. Not that it turned into the industrial production of everything, because of the same difficulties, as usual with the companies like ours, with the funding and waiting and everything else. But basically having the same vaccine protecting human model, not human, but the mice model for human herpes against the HSV2, and the same vaccine protecting chicken against chicken herpes was unheard of. So let me go with this. So those of you who are interested can later look at these two documents. This is basically a little bit of an opinions about us. This is from the Dean of the Medical Cornell, and the left one is from Karolinska Institute, from Professor Goran Hansen, who was for 17 years a member of Nobel Prize Committee and a secretary of the Nobel Prize Committee. And he was also very excited and wanted this work to proceed. Unfortunately, as of today, we're still trying to find the ways to do that. Those of you who live and work in Bay Area probably heard about Peter Farley. Peter Farley was a good friend of our company. Unfortunately, he died 11 years ago. He was a founder of CE-SCV. So these are the people who invented PCR. So he was, and they received the Nobel Prize for that. Peter was a huge fan of our work, mostly because he was interested in connection between herpes and Alzheimer. If you go on Google and you Google herpes and Alzheimer, you will get a lot of very interesting information that was discovered by the wife and a husband team of virologists at the University of Manchester in England. Peter has a YouTube film about us and about our work. And after this presentation, after they will put it all on a different website at the IEEE, we will include the links to these documents and to Peter's film. So what is left now, I think, is to quickly translate our work into into COVID situation. So I believe that using the same principles, either both biological or mathematical, like what we do, we most probably can create a vaccine against all known coronaviruses, known and unknown, hopefully, and their variants, and that way completely change the situation with the RNA viruses. I hope that it can happen, because now most of these years that we were doing this work, the big business was not really interested in virology, was not that interested in immunology. They think that, okay, well, doctors will take care of it for us. Well, now, after they lost zillions of dollars and the pandemic developed and killed enormous amount of people and businesses, probably there will be people who would wake up, and instead of relying on just known labs and known technologies and known universities who were doing it for years and years and years, in a way the same thing, will start looking for more innovative, smaller companies and idiots like us who spend a quarter of a century of developing something that really people don't even pay attention at. But again, then the time comes when it shows what really works and what really doesn't. We know that our stuff works. We're not sure that it works in every case, but it has to be tried, in our opinion. That, and for me, another big target is not only developing just a vaccine for one group of viruses, but another group of viruses, but try to develop the vaccines, several vaccines that will cover several most dangerous groups of viruses. So the new threats that appear, from jungles, so to speak, the new, because of the interaction between different species of animals and all that, you've already heard about what happened probably in Wuhan and other places. And when, you see the Ebola virus suddenly jumping out of African situation, that there will be already vaccines that will take care of that, or at least will partially take care of the severity of the symptoms, and will allow us to engineer, because remember, as I told you, we're using tiny antigens, and we can engineer them and we can tweak them. If you look back to my table here, you will see that each and every one of these antibodies were working against all other herpes viruses, but in a little bit different way. So we can tweak them depending on the genetic of people, depending on mutations and other things. While in today's science, all they do is they just take another huge antigen from a new variant, and for that it has to exist, stick it next to the previous one, and even that is not, I think it's not totally tested yet. So this is for what we can do in 45 minutes, so to speak.
Yeah, I'd like to add
if you have another 5 years, we can, we can.
Yeah. No, when you mentioned the variants, so going back to the SARS-2 and that spike protein I mentioned, so people are probably listening that are concerned about the variants and the changes and such, and a lot of what's happening now is injecting more of the same, hoping to just jazz up your immunity, and there'll be a little cross reactivity and kind of minimize any subsequent sicknesses from other from COVID-19. But the as it begins to mutate further and further, as Alex was saying, by these methodologies, you have to generate a brand new antigen against it. And we're trying to look at something that's more common, that would be a little more long lasting over time.
Yeah. So now we're ready for the questions.
Wonderful. So we have a couple of questions, but I'd like to open up the discussion with my personal question, which I'm very interested. And that is, Alex, who is funding your company? Where are you getting your funding resources from?
That's a very interesting question, and I'm going to give you interesting answer. So we have two companies, right? We know each other because we do the same work. We're not connected in terms of any other ways. It's just hearing about each other for many, many years. For many years, both companies were funded. You were funded a little bit by private investors.
Angels.
We were funded by us. We were just a tiny group of very dedicated people who spent all the money we could, taking it from our families to do that work. That's why it takes. We also, I can put it on my scorecard, is that several years ago we had an investment group coming from a country that I was not really very. You see, the virology is an interesting thing, and you have to be cautious about how you spread the new discoveries in virology and all that stuff. So I was a little bit, I was uneasy about them giving us money and taking control almost immediately. But because cancer is not virology, so to speak, right? And it's not that dangerous, that's my opinion. I invited Mike and his company. We already knew that each other for a long time, and they funded them. So now Mike's group is funded by the group that I kind of dropped for the security reason mostly. And I'm very, very glad that they're progressing, because what they've done is that basically they're now in the first stage of FDA, which means that they're on the cosmodrome. There's a it just lit the fire. Yes. And Mike,
you're on fire, Michael.
But we remain the unfunded group that does everything we could. We did a lot of work recently on the therapeutics for COVID, and then people can go to the websites, our website, or the website of the ResearchGate, and take a look at our work there. You can use my last name or the name Bio-Virus, our company. We are now on the independent scores. We're number 1 and number 2 in the work for lysine treatment for COVID and chikungunya virus. But to answer your question is that no, we don't have adequate funding at all. I think it's ridiculous and I think it's outrageous, but that's the way it is.
Okay, so here's a couple of questions. Why don't the 4 small pieces of labyrinth protein take on a new shape when they are removed from the rest of the labyrinth protein?
Why
don't the 4 small pieces of the labyrinth protein take on a new shape when they are removed from the rest of the labyrinth protein?
Good question. So fortunately, labyrinthine, sometimes you're better to be lucky than good. Labyrinthine is very linear. It's like a rope. And so by being linear, all we have to do is take the different segments based on the amino acid sequence, and we have these algorithms and then biological testing to ensure they're immunoreactive, and we pull them out and we find that they work, at least in the animal models, and it's very linear. It stays open, and then you do the testing to confirm what the question is asking, and we do it in the lab and show that antibodies against the whole labyrinthine also can recognize one of the peptides, and then we have to make other antibodies against the other 3. And we have done that. So that was part of our FDA package to show that all 4 are immune and can cross-react with the whole cancer cell model with labyrinthine there. So
And the second question is the labyrinth protein 100% conserved in one type of adenocarcinoma? What would happen? I don't know the word. It's like, what would happen if you put the whole labyrinth protein in a lab vax? Is the labyrinth protein 100% conserved on one type of adenocarcinoma? And what would happen if you put the whole labyrinth protein in lab vax?
okay, I don't know if there were two questions, or I'll answer the one I just heard.
Go ahead.
The labyrinthine, you could put the whole one in there by increasing by having the proper concentration of the multiple antigens we use. We're facilitating a greater opportunity for a family of antibodies being made.
For the precision antibodies.
Yeah, for precision antibodies being
because
made.
you cannot predict how the immune system will react to the whole labyrinth, which is a huge protein.
And there are some sequences, if you start cherry picking, labyrinthine, that might cross-react with something else that's in your body that's a normal protein, and you don't want that side effect. So we're looking at sequences that are located nowhere else on the human genetics genome.
And I'm sorry to interrupt you. An important thing to say here is that what we are trying to do is engineering. Our vaccine are engineered. We don't just throw things there and see what happens. We try to understand how things are happening and try to combine them in a way that makes more sense and that avoid the negative consequences. Because if you do the cross reaction with the normal human tissues, then your immune system can become autoimmune. I mean, you can get an autoimmune. So throwing a big protein and just hoping that something better will happen is precisely what was done for many, many years. And we're trying to avoid it and trying to avoid the consequences of that.
Alex, please share what lethal dose the mice that all survived were exposed to. What was the lethal dose for mice? And
lethal dose, if you can see in our table, it was LD50. What LD50 means is that it's a dose of the virus that in statistical studies will kill half of the virus in the group. It differs from one model to another. It differs from one virus to another, and it differs from one experiment to another, because you understand what I'm saying? It's as much virus as you need to kill half of the mice group. It was actually invented by Salk when Salk was making the vaccine for, what was the Salk? Polio, for polio. It was a live vaccine, so they had to be very, very careful not to use too much. So he found that if you use half of the, or use on children, half of the dose that will kill half of the rhesus monkeys, everything will be good. Nobody will get sick, but the good immune response will be achieved. So LD50 is that amount, and it changes from virus to virus.
Should we take the COVID booster shot?
No, I'm not going to answer that question. Should we? I'm not going to answer that. I hope you will not.
Should we take the COVID booster shot?
See, the one thing I want to say here is that calling them booster shot is a little bit, in my opinion, in my personal opinion, is misleading, because booster shot is a second shot of the same vaccine. A booster shot is done when your immune response is not strong enough. But when your vaccine is stopped working or working worse against a new variant, it's not a booster, because you're changing the antigen in your what you call booster. It's basically another vaccine, which has to separately be proved to work.
But yeah, that's the good setup. But actually, there's some very serious ramifications for this. So first of all, why not get tested for antibodies anyway, or T cells, other pathways before you get it? And I'm going to tell you why. There's two reasons. Number one, look at it like RPMs in your car. You're already revved up. Is it good to go into the red zone? No. That's basically what we're doing to a lot of people. Number two, if you're already pre-immune, the data are overwhelmingly there with unopposed that if you're already pre-immune, for example, like naturally immune, and you have antibodies and you get vaccinated, you're highly, highly, highly more likely to get side effects and serious adverse events. So if you're already naturally immune, we're doing more harm. It's supposed to be first do no harm, is the Hippocratic Oath, but yet we're doing more harm by vaccinating people who are already immune. That's the other point. Now, finally, for whoever answered this or asked this and anybody else, you could Google antibody dependent enhancement, ADE. And that is a serious concern with all vaccines, but in particular this, because it's basically getting you into the red zone of RPMs, and it can actually facilitate the development of COVID. Remember, SARS is the virus, COVID is the sickness. It can actually facilitate.
We're gonna be crucified for this.
Yeah, but well, the good news is I could just reference all of the papers, no less than about 100 of them.
He is a professor of immunology, okay? So.
Yeah.
So
this
it's a
contradicts the popular opinion and what the social media feeds
We're
us.
clearly not anti-vaxxers. I mean, we develop them.
Right.
So you can't call it, you can't accuse us of that.
When two people develop different vaccines for 25 years, basically for free, you can't call them anti-vaxxers.
No. I'm just referencing the peer-reviewed literature, and it's really unopposed. It's very disappointing as a medical professional. It's not getting out there to the lay public.
Okay, so the question we have is, do you think LabVax is mostly T-cell vaccine rather than B-cell vaccine?
Okay, that's somebody that knows the difference. So B-cell, for the rest of everybody, in short, will be when you vaccinate, you've got cells that make the antibodies, okay? And over time, the antibodies will wane. And even with the COVID, all this mis and disinformation out there. Oh, antibodies are going away. No, they're supposed to. It's called memory cells, okay? And then when you're re-exposed, it comes up. So that's the B cell. And we had a version 1.0 of this vaccine went into patients. And it was basically, you're looking for safety. And we had some interesting success with stage 4 lung cancer patients, developing antibodies, and actually the tumor was shrinking. Now, and they went 2 years beyond their anticipated death date, which is incredible. So it was encouraging, but it was not designed for T cells. So our peptides on this LabVax, I would call it version 2.0. And we've extended our peptides. We've looked at sequences and had to here's your total engineering. We had to re-engineer and look at the structures, if you will, and see which ones would elicit a T cell response. So the current vaccine is a B cell and T cell designed vaccine.
Wow. So it's artificially designed vaccine with a T cell.
Yeah, it's designed to raise B cells and attract T cells as well. Yeah.
Okay.
Dual.
Got it. Next question is Labyrinth 100% conserved in one type? Hold on. Is Labyrinth 100% conserved in one type of adenocarcinoma, say lung cancer? So is it Labyrinth 100% conserved in one type of adenocarcinoma?
Okay. The answer is yes. Now that word conserved is going to be loosely used here because what is unique about this, it's not like any other target that we know of, meaning most targets now already exist in some form in nature. For example, for those that are aware, and they're asking questions, I'm sure they're aware, it's something called Herceptin for breast cancer and others now. That receptor, that target, that antigen is a normally occurring thing, something you need. It's called epidermal growth factor, but it's a receptor, but expressed in large amounts, and aberrantly it's associated with the cancer, just as you could have estrogen with breast or androgen with prostate that are sensitive, but you need those receptors for life. So anyway, that said, labyrinthine is not normally expressed at all. It's a neoantigen. And for some reason, it arises from, from its gene, and we don't know the pathways and mechanisms why it's turned on, and then but we do know There's a protein very similar to it that's inside the cell, locked in there. And this one is missing that targeting sequence, labyrinthine, has lost that anchoring to stay inside the cell, and it pops up to the surface.
Mm-hmm. Got it. Thank you.
It's on all adenocarcinomas, no other cells.
Any other questions, guys? Okay, so there is another one. Okay, could you please explain again why putting the whole labyrinth protein in lab bags would be bad?
Would be best?
Bad.
Oh, no, it wouldn't necessarily be, okay, bad across the board. That's a relative term. We just think putting the antigens, the 4 antigens, would be better because they're more specific. It allows you to create more antibodies against 4 regions. There's less randomness associated with it.
Eliminate randomness.
And I'll give, maybe this would help. If you imagine 255 Lego pieces lined up in a rope, so to speak, 255 amino acids. And we're taking a 22, a 22, a 22, a 23. And it's deliberately cherry-picked to stimulate the immune system. And one other key thing, remember, these cancer patients already have labyrinthine. Then why aren't they reacting the way hopefully we're all healthy, but you're creating cancers conceivably in your body as we sit here.
By the way, the same answer I can give on the herpes side. Herpes is a very difficult group for vaccination. And herpes is a very difficult group. I mean, you know that when people get herpes on their lips, for instance, right?
Right.
In 3 or 4 days, they're gone. So your immune system fought them, right? So why a month later they come back? Because the immune response could be very short-lived. And when you throw a whole, like herpes virus, which is a huge virus, somehow the human immune system doesn't really know how to deal with that. If it did, we would not need the vaccines. Okay, so we know now, for instance, that there is a whole class of multiple sclerosis because people get the people get the herpes infection, and the immune system miscalculates and issues the antibodies that are actually starting to eat the nerve cell myelin. So people get multiple sclerosis, but the reason for that is herpes infection. The same thing as this husband and wife team that I mentioned before in Manchester that you can find by Google, herpes and Alzheimer, is that they found that the long-term coexistence of a herpes, which is a latent virus, with the human system creates that the Alzheimer has a very deep-rooted connection with the herpes virus, and so on and so on and so on. So throwing a bigger model into the immune system is exactly what I was calling a mimicking nature. It works for a certain time for the certain jobs. But if you really have a good bridge, you don't throw a tree across the river like the nature does. You build a bridge, engineer a bridge, learn how to do that and make it to the specification that you want.
Well, and I need to close one loose end. I was thinking about this for that question. Yes, so we want more specificity and a larger response, both. And I left it with that, well, why do cancer patients then not react against this? What makes you think that it'll work with a whole labyrinthine molecule then, because they're not recognizing it? So we want to streamline it with those that would be more recognizable and trigger immune reaction. And also one thing not mentioned was we have a particular adjuvant. So you have something to help wake up the immune system with the vaccine, and then you direct that alerted immune system to what the target is. So we have, an adjuvant is something that'll stimulate the immune system.
And by the way, those who didn't notice that, see, we both use very, very small antigens, and we both achieve universality without actually hoping that much, right? Mm-hmm. It's just we discovered that these small, precise antigens can connect you to universality, because universality is what is common, and big things are not common. It's the small things that are common. And that's why, in my opinion, at least, throwing the labyrinth in there, I mean, it might work at some, but methodologically, it is not what we're doing.
That's right.
So there is a chance that vaccine against herpes can help reduce the incidence of Alzheimer, MS, and other diseases.
Not only Alzheimer, and again, I mean, I would recommend people to read, to get on that your website when we will put the links,
Right.
and to also read to read the work of the, I will put it there, the work of the Manchester University, but also see the short film from YouTube with Peter Farley, because he talks about that in particular. Another connection that is developed now is a connection between herpes and atherosclerosis. I'm not going to waste much time describing it here, but you can also go, and people can go to Google and say atherosclerosis and herpes, and they will get results, and they will even probably find our patent for the anti-atherosclerotic vaccine. Herpes is a virus that is a very elusive, and it's also quite a virus in the ramification of its influence on the human body.
Yeah, thank you. And, oh, there is one more question. Wouldn't the whole labyrinth protein give the immune system more places to react to?
That's common thinking that's been around for a while, right? You know, the larger the protein, the better opportunity for presenting to the immune system. Again, it's already been presented to the immune system, so you have to do something different. It's already there in the cancer patient. So adding, you know, that molecule is redundant in some ways.
Mm-hmm.
So we need to use a different approach for those people.
Should the FDA approval process be different for your vaccines compared to the existing process?
Let me ask that question real fast, because you're a specialist, but I want to throw the real fast one. Because our vaccine can be synthesized, we didn't touch that. Our vaccines do not use viral material. So why I'm talking about our vaccines, because mostly vaccines are viral, right? They're not cancer vaccines. We don't use any viral material. When my algorithm comes with a formula, all I can do is go to the lab, you know, actually put it in a computer, and it will synthesize that peptide. Completely synthetic, no viral material, zero possibility of anything arising from, you know, viruses and viral vaccine or not. And that makes probably half of the concerns of FDA in terms of viral stability, genetic stability, right? You don't have to have a viral lab for that. You don't have to have that 4-level, you know, the virology labs where you have to put enormous amount of effort for the virus not to jump out of the lab, because we don't touch viruses. And
approval will be a lot easier. So it is okay. There is kind of a business / logistic aspect to it as well. I'd also want to mention that there are some sequences in the 255 of labyrinthine we may not want to raise an antibody against. So the 4 that we picked, and we have a few more than 4, but they're not found anywhere when you go to Gene Bank and do a search in any other
genetics.
Exposed, if you will, inside the cell. They're there, but not outside the cell. But if you go of that 255 amino acids, there's some regions there you do not want an antibody. You would prefer not to have an antibody against. Let's put it that way.
Yeah. Okay, guys, we are way over time, but it was very interesting discussion, and we got another question. Considering the huge economic and human costs of Alzheimer's disease, why won't the NIH fund you? Considering the
I can tell you, NIH don't fund things. Peter Farley brought us into CDC in 2007 to the head of the CDC, Kevin Fenton. And Kevin looked at it, he liked it very much. And he said, oh guys, okay, we can help you with the money, but you will have to give us all your discoveries. You will have to open everything. And Peter actually stormed out of that. And we were like, this is, Peter's exact phrase was to Kevin was, this is America, Jack. And you cannot force a private company to give you all the results of which works. Just give it away as a condition for unknown funding, whether you'll carry it to the end or not. So I have all the letters from CDC on that. I have almost non-letters with the words that I would rather not use, coming from Peter Farley, coming from the guy who was a founder of the CETUS, who was discoverer of PCR. And that is because they usually work with a huge pharmaceutical companies who never need any money. They have all the money in the world, and funding things for them is a very unusual thing.
I just, if I could just add to that real quickly, there is other funding besides a second mortgage on your home early on. Anyway, that said, that has to be the story for anything that's going to be successful anyways, your sacrifice. But that said, there's a nice little plot that shows, if you look at NIH or other external grant funding and go bench to bedside or idea to bedside. And it's very high up here overall. Yeah, at any given point, you have 20% odds or whatever getting a grant. That said, the funding is here. And then when you get in the translational phase, it drops. And the NIH did make some adjustments in the 90s to do a little bit of preclinical work, but they still will go much further. And then the other shows, the other y-axis is corporate funding or private funding. And it kicks in. And of course, a lot more goes in because it costs more as you go to phase 3 and phase 4 beyond that. But there's a lot of funding there. So a lot of products are stuck in this traw at the bottom in between public and private funding. And one way around it, we got an NIH on Small Business Innovative Research grant. So there is some funding there, but it wasn't really to go to the clinic. It was actually to go make it you have to have a hypothesis testing. So it's not just, oh, we're going to take a product, we need money to go and make a synthetic peptide and go into patients. They will not fund that. But we did have it for making an improved humanized antibody to attack labyrinthine.
And in general, the funding of these projects is really a completely different discussion, and we can go for hours on this thing. But the point is that even the places like DARPA is really not paying, in my opinion, paying attention. They have preconditions, pre-ideas, their own scientists, their own understanding of things. And in my opinion, instead of looking for the new things, they're looking for confirmation of what they already believe in.
That was personal commentary.
Okay, so I'd like to wrap up this session. It was very interesting and we keep going.
Right.
So I think I want to thank the panelists and presenters. Alex, Michael, thank you very much. It was very informative and I hope the audience will enjoy it. The slides and the presentation will be posted on IEEE Consumer Technology. Also, I want to thank you, IEEE Society in Biotech Medical Consultants with Brian Berg for helping us to promote this event. So I want to thank to Nikki Gastineau for sharing it with the Computer Associates Society. And thank you very much for attending. And I hope to see you next time. And
thank you so much.
Thank you. Bye-bye.
Bye.